Let's jump right into this explainer. Welcome to our rapid-fire Socratic clinical review covering molecular biology and cellular signaling.
From a single, tiny codon all the way up to complex human pathology. Understanding these microscopic processes is absolutely crucial. It's literally the key to unlocking massive systemic diseases.
So, let's start right inside the nucleus with a central dogma. Decoding mRNA to synthesize proteins? That's translation. And here's a strict rule you need to remember. Protein synthesis always physically proceeds in the amino to carboxy terminus direction. We usually just call that N to C. Also, because our genetic code has 64 triplet codons, which way exceeds our 20 basic amino acids, the code is what we call degenerate. Now let's test that knowledge. Moving seamlessly into gene expression, what specific process actually occurs during the translational control of eukaryotes? Think about it. Is it protein degradation, RNA processing, post-translational control, or transcription?
If you guessed protein degradation, you hit the nail on the head. Protein degradation physically falls right under the biological purview of translational and post-translational regulatory pathways. You see, eukaryotic gene expression is controlled at 4 major levels, and degradation is structurally locked into those downstream translational loops.
Okay, let's switch gears to bacteria for a second. How exactly is the tight binding of RNA polymerase to a specific promoter region achieved in prokaryotes?
When we contrast simple and complex systems, it gets really fascinating. Bacteria use these handy accessory proteins known as sigma factors to lock on tight. But eukaryotic transcription, it's a completely different ballgame. It's highly complex, requiring 3 separate RNA polymerases, I, 2, and 3, plus active chromatin remodeling. Because eukaryotic DNA is packaged so tightly with histone and non-histone proteins, it actively has to change structure just to give those polymerases access.
And all of this transcription is happening across the entire human genome, which finalized mapping shows contains an estimated 25,000 to 30,000 active genes. It's kind of crazy to think about, but human DNA sequence mapping is roughly 99.9% identical worldwide. Our personal variance is heavily dictated by single nucleotide polymorphisms, or SNPs.
Let's zoom out a bit and look at the cell cycle machinery. If cyclin-dependent kinases, or CDKs, are the engine, then cyclins function as the gas pedal. They activate the engine to push replication forward. On the flip side, cyclin-dependent kinase inhibitors act as the structural brakes, slamming a halt to replication if any DNA damage is detected. Speaking of that engine running, during which specific cell cycle period is the genomic DNA actually replicated? Does this happen in the S phase, the G1 phase, the M phase, or the G2 phase?
The correct answer is the S phase. That's the dedicated synthetic temporal window where the entire genomic DNA template is duplicated and replicated. The G1 and G2 phases, those are really just gap preparation phases that flank that critical synthetic interval. And the M phase is when the actual physical division occurs.
Now, what happens when things go wrong and those anti-proliferative signals detect damage? In cellular apoptosis, what does the release of cytochrome C actually activate?
Does it trigger the FAS receptor, a membrane-bound death receptor, the TNF receptor, or the caspase cascade?
It's the caspase cascade. When a cell experiences intracellular stress, it causes mitochondrial membrane permeabilization, literally leaking cytochrome C. That leaked cytochrome C then forms the apoptosis complex, which directly triggers the proteolytic caspase cascade, structurally breaking down the cell from the inside out. It's like an organized self-destruct sequence.
Moving outward to the cell surface signaling, here's a classic exception question for you. Which of these is not a classic cell surface transmembrane receptor superfamily?
Is the odd one out transmitter-gated ion channels, GPCRs, enzyme-linked receptors, or adhesive receptors?
The exception here is adhesive receptors. Adhesive receptors, like integrins, exist merely to anchor cell structures to the basement matrix. So they don't belong to the 3 core signaling receptor families that actively receive and transduce external molecular signals. They're structural anchors, not communicators. And what happens when these crucial pathways fail? Pathological dysregulation of TGF-beta signaling is associated with all of these conditions, except which one? Are we looking at malignant oncogenesis, uncomplicated indirect inguinal hernias, Marfan syndrome, or thoracic aortic aneurysms?
The exception is uncomplicated indirect inguinal hernias. Those are purely developmental anomalies. In stark contrast, TGF-beta aberrations actively and dangerously drive oncogenesis, mainly because cancer cells resist its normal growth inhibitory effects. Plus, these aberrations directly drive matrix defects, leading to Marfan syndrome and thoracic aortic aneurysms.
Finally, let's talk about type 2 diabetes. It's actually completely false to blame direct insulin receptor mutations for this disease. While there are some very rare syndromic variants out there, the vast majority of standard clinical cases result from progressive downstream post-receptor signaling defects. That's what makes it such a complex, polygenic metabolic disorder.
It really is wild to think that a single misread codon or a progressive signaling block is where massive systemic disease begins. It leaves us with a fascinating question to ponder. What other microscopic errors are actively shaping human pathology right this very second?